Well, first I'd like to thank Michael Buchmeier and Elaine Pedersen for having me here at this great symposium, and we're really looking forward to the afternoon too. It's awesome to learn about the RNA science here at UCLI. What I'd like to do today is talk to you about roles for liver-specific microRNA 1 to 2 during hepatitis C virus infection. So I will tell you how the virus subverts this microRNA. Subverting means you take it and you use it for a very different usage as it normally does. And I will tell you it's Achilles heel for the virus. We get rid of the microRNA, viral RNA gets lost, and I will show you at the very end that ours is taken away into the clinic. Let me introduce you first briefly to hepatitis C virus RNA. It's a positive stranded RNA virus, so it's about 10,000 nucleotides in length. It's a very 5' end structure, and it contains an iris and non-ribosome entry sites. So it functions very much like a Scheinberg-Gardner sequence, where you can bind directly for the subunits and start translation here at the very 5' end, starting with the core protein. And the RNA is translated with the polyprotein, about 220 kilodalton in size. And at the end terminus we have the structural proteins, and then the C terminus we have the non-structural proteins. 3' end is very conserved, it's structural, and it does not have a poly-A tail. Now, many people are infected worldwide, and of course it takes about 30 years before you really know you're infected with hepatitis C virus and your liver really gets damaged, and therefore people who maybe 30 or 40 years ago received bad blood during surgery, they are infected now. And of course today you only can get the virus if you share a needle or if you get bad blood somewhere in the world. It's not sexually transmitted. We know a lot about these viruses. We know there are 6 different genotypes, and depending where you live, certain genotypes are prevalent. Now, the good news really is we have a drug, sofosbuvir, which really can cure hepatitis C virus. The bad news is it's very expensive. It costs $80,000. You have to take 80 pills. Each pill costs $1,000. But again, there is a good news coming again, because Gilead, which has manufactured this drug, have lowered their price, and as soon as they lowered their price, their stock went up. So I think it was good for both sides. So what we discovered about 10 years ago, at the very 5' end, there was a sequence motif, and the sequence motif seemed to predict, and we heard about it from Ian this morning, that this sequence could bind 2 molecules of microRNA 1 to 2, which is highly abundant in the liver. And we were intrigued by this because it's the very 5' end, and as you remember from talks this morning, microRNA seem to interact with 3' non-coding region, or Irene has told us maybe even more. But it was really not known whether microRNAs can or do bind to the very, very 5' region of an RNA molecule. Of course, we thought, well, microRNAs repress gene expression, maybe it binds here, represses gene expression. But again, it was odd, and it was a very, very 5' end of the viral RNA genome. The other curious thing was that microRNA 1 is highly, highly abundant in the liver. So usually in the infected liver, we have 1 to 10 molecules of hepatitis C virus RNA. I'm talking about infected human liver. And there are about 66,000 copies of the microRNA in the liver cell. So you can imagine this 10 molecules of HCV RNA are there, surrounded by the sea of this microRNA. And if the virus really, as I predicted on the last slide, binds this microRNA, we thought maybe this has a very important function in the life cycle. So the first question we wanted to ask, of course, is this microRNA important for hepatitis C virus? So the first thing you do is you get rid of the microRNA and ask what happens to hepatitis C virus RNA growth. So what you can do is, here's the microRNA, you can sequester this microRNA with the so-called antisense LNA locked nucleic acids. Locked nucleic acids have a ribose, a very interesting ribose, in which the 2' and 4' position is linked by an oxymethyl group. So as a result, the ribose is a little bit in a different conformation. And even if you have only a 15 neuron, and shown here the LNA, shown here in green, it will very, very tightly bind the microRNA, not only in tissue culture cells, but also in the liver of an animal, and as I'll show you later on, in humans. And the tiny little duplex is not really big enough to really activate all kinds of innate immune responses. It does not at all. And the other thing is it will sequester the microRNA very tightly, and in the liver in an animal for about 7 weeks, and then after 7 weeks it gets secreted via the kidney. So it's a very temporal, very highly efficient sequestration of the microRNA. So what we did here, this is another plot, we basically infected cultured H8 cell peptide C virus, and we added the RNA, and as you can see here, a very, very low concentration, 0.5 nanomolar, and this is unformulated, we just add it to the medium. It goes into the cell, and you see a drop in hepatitis C virus RNA bindings. So of course, then we wanted to know, is this a direct effect? Because I just showed you that if RNA sequesters microRNA, you want to use HCV RNA, but this is because it doesn't interact anymore with HCV RNA. So what we can do, of course, we can synthesize mutated or wild-type hepatitis C virus RNA in vitro with the 7-RNA polymerase. And what we can do is we can insert mutations in the predicted binding sites for microRNA 1 to 2. So if you make a mutation in binding site 1 or binding site 2, shown here, transfect this mutated RNA, no viral RNA is being replicated, no viral RNA abundance is being noticed. However, if you express topically a mutated microRNA that now is predicted to bind to the mutated genome, we can rescue a hepatitis C virus RNA abundance here at site 1 and here at site 2. Then you might want to ask the question, well, do both of these microRNAs need to bind at the same time? So basically, you make a mutation here, make a mutation here, and this shows you can only rescue hepatitis C virus RNA abundance if you have both microRNAs bound to the same RNA. So here we have a model how this, we did of course a lot of mutagenesis on this complex. Here we have microRNA found at site 1 and found at site 2. And what you can see here in these boxes in red and blue, that these sequences are absolutely highly conserved among all 7, I only showed you 3, among all 7 genotypes. So if you go to the gene bank, there are about 10 to 15,000 sequences of HCV sequences from HCV patients. You will never ever find any mutation in these sequences, which is very unusual because, as you know, RNA virus, they make a lot of mutations because the RNA-dependent RNA polymerase error makes a lot of mutations. But these sequences are highly conserved, they never ever change. And so what I want you to pay attention to is nucleotide number 3, the HCV genome. Is a C here, okay, and he base pairs with the microRNA 1 to 2 at site 1 with the G. Just remember this because at the very end of my talk, we will come back to this. So this nucleotide here, 1, 2, 3, C, seems to interact with G and it's very highly conserved. Alright, so we have this complex and we identified it and we asked the question, so what's the mechanism? By what mechanism does 1 to 2 enhance HCV RNA bindings? You can think about translation, replication, RNA turnover. And it turns out that it's actually RNA turnover. So it really protects the viral RNA. So what we do know is that a hepatitis C virus RNA genome contains a triphosphate. Now, as you know, if you have a triphosphate RNA in a mammalian cell, you activate RIG-I because there are no molecule in mammalian cells, with very few exceptions, small ones, that have a triphosphate. So RIG-I is being activated. Of course, the cell will turn on a lot of the stimulated genes and go into apoptosis. So we thought, well, this is cool. Maybe this RNA is acting like a shield. MicroRNA acts like a shield. And it protects probably the triphosphate from being sensed by RIG-I. While this sounds reasonable, it might still work and have an important role in the liver. We know that in culture cells it's not important because we have cells that are defective in RIG-I and it still needs microRNA 1 to 2. But also we thought, well, maybe we should look at the nucleases, at 5' to 3' nucleases that operate in the cells. And there are 2 nucleases in cells. One is exon 1 and one is exon 2. Exon 1, and of course we were biased, this is the important one, is in the cytoplasm. And it's a so-called processing bodies in the cytoplasm. So if you do have an RNA molecule that's being translated and the cell is stressed, let's say, by a variety of means, infected with a virus or there are no nutrition available, the RNA is not being translated, it gets put into this processing bodies there, it gets decapped, and when it's decapped it has a 5' monophosphate, and exon 1 will degrade the RNA. Now,
there's also an exon 2 nuclease, very similar, and that one is in the nucleus. And I just want to tell you, because it's neat, not many people know about this. So when Pol2 transcribes DNA, you make a transcript, and as you know, finally you hit the AAU-AAA poly-A signal. 30 nucleotides further downstream, you add a poly-A tail. And this is your mRNA, it comes out of the nucleus. However, Pol2 continues transcribing the DNA. Now at this point, of course, because the RNA is being cut off and the poly-A tail has been added, there will be a 5' phosphate. So exon 2 in the nucleus actually attacks these RNA molecules and tries to catch up with Pol2. And as soon as it catches up with Pol2 will fall off. And that's the transcription termination is really caused by exon 2. So as I said, of course, the HCV is a cytoplasmic RNA virus. So of course, we assume it would be exon 1 that might detect a hepatitis C virus RNA, and microRNA 1 to 2 might be a shield. So however, it was not the case, and because this is a complicated species line, but basically what we do here is we are knocking down with siRNAs a variety of nucleases and components in the processing body. But if you look here at exon 1, you see we can efficiently knock it down, but there is no or very little effect on, for example, on the hepatitis N-spike protein or core protein. Well, so at that point we had to pay attention to exon 2. So as I told you, it's in the nucleus. Well, is it in the cytoplasm? It is in the cytoplasm, not as much, but it clearly is. And so what we noticed already, so that it is in the cytoplasm, but it doesn't matter whether the cell is uninfected or infected with hepatitis C virus. So what we did is, of course, we depleted exon 2 by siRNAs, and when we do this, we see a tremendous increase in hepatitis C virus non-abundance. So that was the first hint that maybe exon 2 is degrading hepatitis C virus RNA. And of course, if you knock it down, you enhance hepatitis C virus non-abundance. Not just HCV abundance, but also hepatitis C virus protein abundance are getting enhanced when you knock down exon 2. And of course, you would expect when you overexpress exon 2, you should see the opposite. Then you should see more of hepatitis C virus RNA loss. And this is indeed the case. If you overexpress and flag tag exon 2, you see there's less hepatitis C virus RNA in the cell. Now, there was a problem. The problem is, I told you HCV has a triphosphate. And I told you also exon 1 and exon 2 are proposed to degrade 5' monophosphorylated RNAs. So that was a conundrum. And so we went back to the original literature of exon 2. Exon 2 was discovered in the 70s by Audrey Stevens at the Oakville National Laboratories. And she showed actually in her paper that exon 2 can also degrade triphosphate containing RNAs. So we tested this. So what we do here is we have hepatitis C virus RNA, it's a real biochemical experiment in the test tube. We have hepatitis C virus RNA, and we add a cap structure, which is of course totally unnatural, a triphosphate, which should be the natural, and or monophosphate, because that should be our positive control. So as you can see, if you don't add any microRNA, just take these RNA molecules and incubate the RNAs with recombinant purified exon 2. You see if you have a cap structure, the RNA is very, very stable. If you have a triphosphate, you see some degradation. But if you have a monophosphate, it gets degraded very quickly. Now, of course, we wanted to ask the question, does microRNA want to protect hepatitis C virus RNA when we add exon 2? And that's shown here in the middle panel. And it doesn't matter if you have a cap structure, a triphosphate, or even a monophosphate, 1 to 2 totally protects it from degradation. And here's a control, it's not just we add a microRNA and then things happen like that. If you have a control microRNA 1 to 4, which is not predicted to bind to HCV, you see the same pattern as you see when you do not add any microRNA. So now the question was, okay, the final experiment would be, well, if exon 2 degrades hepatitis C virus RNA, then when exon 2 is not there anymore, HCV should not need 1 to 2 to be protected. Right? And indeed, this is the case. It's a complicated experiment. But what we do here is we add siRNAs against exon 2, and I showed you before hepatitis C virus RNA abundance goes up. So this is the normal, and here's a quantitation. And to our delight, of course, we found that when we sequester microRNA 1 to 2 with LNAs, then we deplete exon 2, also hepatitis C virus RNA goes up. So here, 1 to 2 is sequestered, but we see stimulation of hepatitis C virus RNA, but only when exon 2 is not there anymore. So that's a pretty cool genetic evidence that 1 to 2 binds to HCV RNA and protects it from being degraded by exon 2. So this is just a brief model here, and so I told you exon 2 is involved in degradation of RNAs made by orthogonal nucleus. In the cytoplasm, we think it will attack triphosphate containing HCV RNA. Of course, we can also imagine that maybe there are helicases, and at steady state, some molecules of HCV do not have microRNA 1 to 2. They might get degraded. And of course, there are some anecdotal evidence, maybe there are some elusive pyrophosphatases in the cytoplasm of mammalian cells. So if this is the case, of course, then we should end up with the HCV RNA molecule monophosphate, and that should get degraded as well. So of course, now we're very excited. 1 to 2 is an Achilles heel for HCV. And the question was, well, what is its normal function? I told you it's highly abundant in the liver, so we needed to know that. And at that time, when we were contemplating about this, that was at the end of 2006, 2 papers came out, one by Sarnow at Rockefeller, and one headed by Christine Azar. At that time, she was at Long Island here in Southern California. And what they did is they inhibited microRNA 1 to 2, not by LNA, but by other antisense molecules. And they could show 1 to 2 was depleted in the liver of mice, in this case. And the animals were fine. They made it, they ate, everything was perfect with them. So eventually they, of course, sacrificed the mice, they cut out the liver, and they did a microarray experiment. And of course they find what all of you find when you do this experiment: 300 genes were up and 300 genes were down. And of course at that point, you look at the list, and I call this the shiny pebble syndrome, because you look at the list and you always find that a gene you're very, very excited about. And of course in the end it's like, it's never the right one. But of course you can look, is there a cluster of genes which maybe function in a particular metabolism, either up or down regulated. And that was interesting to look at this cluster. Does anyone know what kind of metabolism is regulated by these enzymes, H, M, G, C, R, scoline, and so on? Cholesterol, right? So, if you sequester microRNA 1 to 2 in the liver, and it's not only in the mouse, it turns out also in humans, then what you find is cholesterol biosynthesis is downregulated. That means the normal function of 1 to 2 is to upregulate cholesterol biosynthesis. And indeed, if your microRNA 1 to 2 is being sequestered by RNAs, your cholesterol level will go down in your blood. And so that was the normal function of it. And so we thought, well, so by what mechanism, what is the target of 1 to 2? So we thought it should regulate, of course, a suppressor of cholesterol biosynthesis. Downregulation of a suppressor which enhances cholesterol biosynthesis. And then we were stuck, what is a suppressor? Because we looked for canonical and non-canonical binding sites, and we just didn't know what to look for. So eventually we went to the literature, and we studied papers mostly written by Brown and Goldstein. The cool thing is, if any of you ever work with cholesterol biosynthesis, you only have to read papers of Brown and Goldstein, and of course the people who work with them, because they have done everything. So that was good. So we came across this paper, and this paper says overexpression of INSIG1 in the livers of transgenic mice inhibits SREBP processing. SREBP is a transcription factor that controls the transcription of all of these genes shown here. So that is very interesting, because what we found, figure 3 in this paper, lists exactly these genes which were in table 1 in the ASO paper. So that told us that probably INSIG1, which is known to be a suppressor of cholesterol biosynthesis, is probably a target for microRNA 1 to 2. And this is indeed the case. I don't want to go through the data, I just want to summarize to you what we found. We found that INSIG1 is made in 3 forms, and the forms only diverged in their 3' non-coding regions. So you make a 1.4 kb, a 1.9, and 3 kb in sequence form. And interestingly, when you look which of these mRNA is actually translated most efficiently, it's actually this one. Because if you have a short 3' non-coding region, in general, the RNA is translated better because, of course, you have by chance fewer microRNA binding sites, of course, and also you have fewer sites to which protein repressors can bind. So we found indeed this mRNA is highly translated, although it's not the highly abundant mRNA available. And what we found that microRNA 1 to 2, while it can bind to other parts in the 3' non-coding region by non-canonical interactions, that's why we have never been discovered it. But what we found that there is a site here that overlaps the poly-A signal that generates the 1.4 kb mRNA. And what we found is it's in the nucleus, of course, where you have polyadenylation, so it's not the mature microRNA. It's the precursor RNA of microRNA 1 to 2 that binds in the nucleus to the poly-A site and inhibits its formation. So therefore, you only make these two molecules, the 3 kb and the 1.9, they're very poorly translated. So microRNA 1 to 2 regulates the expression of INSYG1 by using its precursor microRNA in the nucleus to modulate polyadenylation usage. So I just want to give you this piece of information because suddenly we were forced to think, what do we know about cholesterol biosynthesis, precursor microRNAs, and hepatitis C virus RNA? Is there any connection that is important? Because we knew hepatitis C virus RNA, hepatitis C virus, and many other RNA viruses love cholesterol. So usually in infected cells, cholesterol biosynthesis goes up because you have more fluid membranes and it makes much more fluid replication complexes on cellular membranes. So we wonder whether there was a connection. So we went to the literature and we found cool, actually, cholesterol biosynthesis is regulated in a circadian manner. First, I was confused by this, so you need to know I've never worked with animals. So I was confused that during the dark cycle in rats in this case, I was confused that cholesterol synthesis goes up. And I asked people, it doesn't make sense. Shouldn't cholesterol biosynthesis go up after you eat? And they say, well, what happens with mice and rats, of course, when you turn off the light and they become active and then they eat. So for us, it's exactly the opposite. In our circadian rhythm in the liver, it's when it's light and we eat and then we're active, cholesterol biosynthesis is up. So then at that time came something, an incredible paper came out by Ulrich Schiffer and Gatfield. And they had actually studied cholesterol biosynthesis in the mouse. And they studied whether the microRNA 1 to 2 is involved in its regulation. So at the same time as I told you, we did this INSYG1 studies. And what they found, which was very interesting to our surprise, that the precursor microRNA 1 to 2 and the primary microRNA 1 to 2 is regulated in a circadian manner, but not the mature microRNA 1 to 2. So that was puzzling to us. So I showed you that the mature one, of course, regulates HCV. But would that mean that maybe the precursor microRNA, which of course upregulates cholesterol, which is good for the virus, but could also the precursor microRNA regulate also hepatitis C virus RNA abundance? And maybe in a circadian manner, who knows? And then, of course, if this is true, that might be important information for drug. So is there any evidence at that point, we asked, is there any evidence for the existence of functional precursor microRNA risk complexes? And of course, Ian showed us crystal structures of Argonaut and microRNA. But he showed, of course, he focused on the 5' end of the first 10 to 60 nucleotides. But actually, it has been shown the precursor microRNA, which of course the 5' end is the same, can be attached to it can be attached to Argonauts. The 3' end of the precursor microRNA simply sticks out. That's also shown in this paper by Tane et al. They found that indeed that endogenous in vivo human Argonaut 2 can bind to precursor microRNA independently of diatom, and these complexes are actually functional. They can function as microRNA or as siRNA complexes. So we asked the question, okay, does precursor microRNA not only regulate NC1, does it actually regulate the abundance of hepatitis C virus RNA? So how do we do this? So we needed to synthesize precursor microRNA that cannot be cleaved by dicer. And at that point, we weren't quite sure how to do this. So we went into collaboration with Stephanie Mortimer and Jennifer Gautner at UC Berkeley. And they gave us the idea, well, maybe what we should do, here's a precursor microRNA, here's dicer, and here's cleavage. You see in green the mature microRNA. We should add ENTPs into this region, and dicer apparently is not supposed to cleave ENTP substrates on each substrates. So we added 9 nucleotides here, or we added 12 nucleotides, or we made a whole ENTP loop. So are these molecules being cleaved by dicer? So again, this data was all obtained by Stephanie. So what she did is she used this precursor which she synthesized in vitro, incubated them with dicer and TRBP, that's a cofactor of dicer, it's a binding protein. And what you can see here in this assay, this in vitro assay, indeed, if you have a precursor microRNA, it's being cleaved, but if you have the DNTPs in there, it's cleaved very, very, very poorly. So now, of course, we ask the question, it's cleaved poorly, cool. Do they function as siRNAs? That's the easiest way for us to really look whether they are functional, because we see also the cleavage product. It's not like you look at the luciferase going up or down. So here we have a GNP molecule, which is a perfect binding site to microRNA 1 to 2. And now we add our precursor microRNAs and ask the question, are they being cleaved? And if they're being cleaved, you will see the cleaved GNP over here. And indeed, what you can see is that the precursor ENTPs, the precursor microRNAs which contain ENTPs, function indeed as siRNAs. Well, do they function in protecting or in enhancing the RNA abundance of hepatitis C virus? And the answer was yes and no. Well, the pre-DNTPs and the pre-DN12 did enhance hepatitis C virus RNA abundance. But when we had a precursor microRNA with an entire DNTP loop, it did not. So there is a caveat, and you can say, well, maybe these ones are cleaved just a little bit, and so that's why it enhances HCV RNA. And this one is not cleaved at all, so that's because the precursor microRNA it has no function in HCV. So we were really stuck because we really needed to prove this. And then we got lucky here, a lucky break in that we got a genetic tool to answer this question. We got a genetic tool by accident when we made a lot of mutations in microRNA 1 to 2. That's a duplex which we usually use to deliver into cells. What we did is we introduced the 2 mutations here. So we basically had a microRNA 1 to 2 duplex which we knew functioned as an siRNA. So we can put it into cells, have a proper reporter, and we know it works very well. But it did not rescue hepatitis C virus RNA. And the reason is the wrong strand gets incorporated into it, so we do that. And then we thought, well, this is cool, maybe we should use this RNA as a baseline and make the precursor of it. Because if it would get cleaved, it would generate this molecule which does not work. And that's indeed what we did is, Stephanie again made this RNA, and she could show that indeed it is being cleaved. But remember, when it's cleaved, it doesn't enhance hepatitis C virus RNA abundance. To our delight, we found that this particular molecule, when it is as a precursor form, it can enhance hepatitis C virus RNA abundance. But when it is in its mature duplex form, it cannot. So this is to us showed us clearly that the precursor microRNA is not only functioning in sync mRNA modulation, regulation of expression by polyA site selection, but also in the cytoplasm can enhance hepatitis C virus RNA abundance. So that might be very important when we think about treatment and when we should treat patients with hepatitis C virus. So finally, I want to tell you with all of this information out there, of course, as I told you, we have sofosbuvir, so we have a great drug, but who knows, 8,000 people have been tested, there are 200 million. Maybe there are some people who cannot take the drug for whatever reasons. And personally, I always feel like we don't have a vaccine, forget about this, but we should always continue to look for other targets. Those targets, we should look for natural products, not just synthesizing something chemically, spending millions and millions of dollars. Maybe some stuff is already out there in the soil or somewhere in plants. We should continue to find other molecules that might be helpful. So, of course, in this case, the question was, can RNAs directed against microRNA 1 lower HCV yield in infected patients? And the answer was, of course, you will go through phase 1 clinical trials, phase 2 clinical trials, everything sounded good and nobody fell down. And eventually, the study was published in March of 2013, where a group headed by Janssen, it's a group in Denmark, also in Paris, they did a phase 2 clinical trial where they treated hepatitis C virus patients with RNAs directed against microRNA 2. And I just want to summarize for you the data. So they had 21 naive patients were enrolled. Naive means these were patients who had never ever received any antiviral therapy. And the dosing was 5 milligrams per kilogram RNA once a week for 4 weeks, and then this drug. And then they looked what happens to these people 10 weeks after the last dosing. No adverse effect. The virus titer dropped 2 to 3 logs in all 9 of patients who received the RNA. And 4 out of 9 patients failed to yield detectable HCV RNA. At that point, so 10 weeks later, they did deep sequencing, there was no other breakthrough. And of course, in comparison, we knew that already. Protease inhibitors such as Glacovil, we saw already viral breakthrough 7 days after it. So now, remember I told you at the beginning of my talk, HCV nucleotide number 3, that C, base pairs, the G base pairs with the C microRNA 2. Now, we are 2 years later, and of course people can go back to these patients and ask what happens, because not everybody got cured. And so people re-isolate 2 years later a hepatitis C virus RNA and ask what happens to them, what happens to the virus, and they do deep sequencing. And now what they find in all of these patients that still have virus left, this nucleotide number 3, the G in HCV, changed to a U. And so that's interesting because it should still base pair with the G in microRNA 1 to 2. But so we are really at loss. We really need to find out what happens. That just tells you know nothing about RNA structure. Is it a protein binding site or is the complex really different? So we are really very hard studying right now with in vivo shape analysis and stuff. How can this nucleotide change, which you would predict should have no effect whatsoever on this complex RNA structure, how can that confer resistance? But that's something we are going to do. And now, maybe I should talk to the students now. As a rule, you should never end a talk with a caveat. But if you have a story and there is a caveat, so never ever put it under the rug. So while we were doing this studies, and you probably have been noticing there's one piece of experimentation has been missing, and that is what's the phenotype of a transgenic mouse that doesn't have microRNA 1 to 2. And of course, I've been talking to these people, to the industry, and I said, make a transgenic mouse because it's very expensive for me to make, right? And they said, why would we do this? Look at this. We don't want to know. Whatever. What do we learn from this, right? But finally, two papers came out last year. One by Kalpana Ghoshal and one by Andy Chu. Basically, you make a transgenic mouse, and the mouse lives, but the mouse develops very early on liver cancer and metabolic changes in the liver, and they really have developed early on carcinogenesis. So, none of us is born or has been born without microRNA 1 to 2, I'm pretty sure. So if you're born with microRNA 1 to 2, we know from animal and phase 1 and phase 2 clinical studies, you can sequester it at least for 7 weeks. But based on this result, none of us would ever suggest we should sequester microRNA 1 to 2 for long term. because it could have long-term effects on the liver, because that's what we know from the studies. So it's always good to keep this in mind. Of course, microRNA 1 to 2 is, as I told you, upregulates cholesterol biosynthesis. Remember, I told you that 300 genes going up and 300 genes going down. Of course, some of them are predicted oncogenes, so we have to be careful. But so far, what we know, 4 weeks dose of an LNA can suppress the 1 to 2 for 7 weeks, and it's out of your system. And during that time, you probably will lose HCV. So if I would be a patient who's not responding to anything, I probably would say, okay, I'll take this 1 dose. But again, it's not a long-term treatment. In summary, I just briefly want to tell you, I told you microRNAs, they do not only repress, they can also positively regulate. In this case, it's a replicating viral RNA. That's important, it's a replicating RNA, it's not an mRNA. There's no evidence yet that an mRNA is positively regulated by a microRNA via binding to its 5' end. And maybe it's not that important, because as you know, mRNAs have captured the 5' end. And it's you subverts it, and again, it makes sense. You have few hepatitis C virus RNA molecule that's embedded and surrounded by a sea of 1 to 2, and probably simply evolved very quickly because it's so bound to binding. And we think one of its functions is, and there might be more, we don't know yet, of course, one is it protects it from being degraded. And I told you the precursor of 1 to 2 has an interesting function too, not only regulates viral RNA, but also cellular mRNA gene regulation. And I just told you LNAs show tremendous promise in clinical trials. Of course, this is the most important slide. We've credits to the people who've done the work I talked to you about. And specifically, Selena Sagan and Erica Mackley, they have done most of the mutational studies on microRNA. Cecilia Sarnow, she did all the work on exon 2. And Carla Parker did the work on exon 1. And Christian Chen, she did a lot of the work on the precursor microRNA for I talked about in the end. And all the cholesterol and the insulin work has been done by Sarnow. And I'll stop here, and of course, I'm happy to take questions. Thank you. So again, this exon 1 is right. Yeah, he asked about the exon 1 or exon 2, whether it's specific for the monophosphate. So what we find is it's most monophosphate containing mRNA is most easily degraded. And if you have a triphosphate, it's more resistant, but it still gets degraded. Right. Yes, exon Y is exactly the same. I didn't show in vitro data with exon 1. So what is the XR target? You showed the XR2 when you knocked it out. Right. I don't know. We can only imagine that it's in the processing bodies and it's probably not accessible. Yeah, yeah. In other words, is there? Right, is there pyrophosphate? I don't know. We really don't know. Yeah.
So it's intriguing that you've shown regulatory functions of a precursor microRNA for both a cellular endogenous protein and viral genome. But the overwhelming majority of microRNA 122 is mature, and the mouse knocked out everything. So I wonder if you could speak to whether it's really true that precursor microRNA is important for liver cell function, or the mature microRNA 122 does something in addition, And if that's the case, then the locked nucleic acid strategy could possibly be modified to be specific for the precursor microRNA function.
Right. So what is, yeah, this is a good question. So what we do know is that the precursor is made in a circadian manner, and the mature one is very stable. It's in the cell for up to 3 weeks. It's very, very stable. So we think maybe there are subtle differences in their functions. We know there are very different isoforms of the mature microRNA, but they are not inherited in the precursor. So our hypothesis is they have distinct different functions, not only because of their half-life, but also because of their isoforms and sequence composition. But it's really difficult to solve these things out. We have started to begin to isolate from different compartments precursor mature microRNAs, do deep sequencing, and so we're getting a hint that this diversity actually exists, but we don't know whether it's functional.
So what is known about stabilizing HCV negative strand RNA? Is there a similar microRNA mechanism in play, or is that not necessary during the viral life cycle because so much less template is necessary?
Right. There is no evidence that the minor strand binds microRNAs. And actually, it's a really cool experiment that gives you the data. And that's done by Joe Luna and Charlie Rice, I think a month ago, and published in Cell. What they did do is they did an Argonaut clip assay in uninfected and HCV-infected cells. Remember what Ian said, when Argonaut gets cross-linked, then you have a good hint, this is a microRNA binding site. So when they did this experiment, they saw microRNA 1 to 2 being bound, at the 5' and when they looked at the minor strand, there was no Argonaut clip whatsoever. So there is no evidence, and I like that data that the minor strand is intact or needs to be protected.
You mentioned that this microRNA is normally function as cytokines, right? But you showed that it can actually go into the nucleus and bind the precursor line.
No, that's the precursor microRNA.
Yeah. So you mentioned that it binds to the RNA signal region. So does that occur in the nucleus?
Right.
And then that prevents the usage of that site.
That's right.
Does that RNA
It's not being made. Yeah. You just, what happens is when the microRNA, the precursor microRNA sits there, the site is not being used, and you use the next one.
Yeah, but supposedly it should have a carbon application.
Actually, that's a good point. We do not know that. Actually, I don't think so. I think probably the micro. Well, I don't know the answer. Yeah, I don't know. Yeah, it is. That's correct. It can be done. I don't know.
All right, let's thank all the speakers for the one.